Okay, I think we are ready to begin. Dr. Stark, if you want to take it away.
Yeah, good afternoon, everyone. Thank you so much for joining us today. We are super excited to be here with our first All-Star Rec performance. This is a pilot project to see if the winners of our poster presentation enjoy this opportunity to share their work with y'all across the country. So let us know how you like this. Give us some feedback. We are really enthusiastic about our 4 Rising Star winners today. I hope you all take a moment to read some of their papers and get to know their work. And hopefully, our whole big goal for this is that you guys start collaborating together. As the future of Alzheimer's disease research, we are counting on you to begin to build connections with each other. This is one step. So I'm going to not take any more time. I'm going to make sure that you all get to spend a lot of time hearing about your colleagues. Welcome, and we hope you enjoy this.
Wonderful. Thanks, Susie. So our first Rec Rising Star presenter today is Quinton Cotton. Dr. Quinton Cotton is an assistant professor at the University of Pittsburgh School of Social Work. And they are presenting today on the qualitative analysis of dementia caregivers' experience with lucidity. Go ahead, Dr. Cotton.
Afternoon. Thank you. I want to thank NAC for the invitation to share preliminary research findings on the topic of lucidity. My funding sources and disclosures are provided on this slide. According to the National Institute on Aging, even in late-stage dementia, patients reported to exhibit unexpected episodes of mental clarity or lucidity, characterized by spontaneous, meaningful, and relevant communication at a time when the capacity for coherent speech has presumably been lost. NIA released a funding announcement to examine lucidity. This slide contains information on potential areas of study to better understand lucidity, and we also see the list of awarded projects here. Data in this presentation is from the exploratory or the R21 phase of the study and comes from the Mayo Clinic Rochester study. The goal of the study is to examine dementia family caregivers' perspectives and experiences with lucid events. With recruitment support from US Against Alzheimer's, we surveyed 233 dementia caregivers. 24 dementia caregivers completed telephone interviews. We will report on the qualitative portion today. Data were quoted by the Mayo Clinic Rochester team, and my team at the University of Minnesota analyzed data for the work presented today. Here is a brief snapshot of survey results. Most lucid events lasted 30 minutes and under. With respect to communication, half of persons living with dementia made complete sense when communicating during a lucid event. For over 1/3 of lucid events, caregivers reported that the person living with dementia had a visit by a family or friend leading up to the event. And another 1/3 of caregivers reported that there were no special circumstances surrounding the lucid event. Now let's look at preliminary results. This slide provides information on lucid event types and lucid event experiences as a process based on interview data. Let's begin with the right side. Lucid events were mostly described by dementia caregivers as singular events, with repeated lucid events reported as rare. Caregivers reported that lucid events were spontaneous or generative. For generative events, having familiar people around was the most common condition leading up to the lucid event. On the left side, we look at the lucid event experience, which includes event onset, initial appraisal, experiencing the lucid event, event termination, sense making and actions taken, event recreation attempts, and long-term appraisal. On this slide, we highlight a positive and a negative experience. In the first example, food was described as a trigger. The caregivers and others were surprised by the person with dementia's verbal communication. He took one bite, looked at me, and said my name. Everybody in the place dropped. They just stopped what they were doing and said, "Did that really come out of him?" It brought me to my knees. It brought me to tears. In the second example, the caregiver reports on their response to the lucid event. Additionally, this caregiver does something that's very important that other caregivers in our sample also reported. The person living with dementia provides insight into their own level of awareness about their condition and/or the impact of that condition on others. She said my name. "I know this is really hard for you. And I know this is going to be much harder for you than it's going to be for me. And I'm so sorry." And I just had to pull over. It was horrible for me, stating the caregiver, because just having your mom apologizing for being sick was just brutal. One caregiver reported coming to terms with the progressive nature of dementia, reporting that they understood their loved one to be gone. Having this information and then having experienced a lucid event was described as challenging. While some people think these events are positive for caregivers, that may not always be the case for all caregivers. Then she was already that gone. And that was a really hard moment. I know some people think it's a gift, and maybe it is, but it is, and it isn't, you know? Caregivers also reported that lucid events resulted in change decisions regarding frequency, level, and setting of their engagement. I felt that we could take him out in the car and try getting him out for a time. And so I guess we would get maybe books or puzzles that would kind of stimulate her mind. So we tried to turn to TV shows that we thought she might like. Caregivers even reported that the relationship between the lucid event and care decisions was important, describing a nursing home potential placement. And I said, "No, she remembers some things. As long as she can remember us, we can take care of us. We can take care of her." Caregivers also reported sort of mixed reactions thinking about their long-term appraisal of lucid events. For some caregivers, the overall experience made them sad, especially concerning ups and downs. And that was the last time he recognized me. It still makes me sad. For other dementia caregivers, they found positive things to hold on to. And it keeps me going now, even 5 years later. I wanted to share one final quote to discuss the importance of always honoring personhood of a person living with dementia. Never give up on them because they're still in there. So even if maybe they don't seem like they're having a lucid moment, just keep on talking and maybe something will resonate. That's what I would say. And this slide just is a concluding slide that caregivers really reported on these moments in great detail that they will never forget. Caregivers truly spoke about their knowledge of their loved one, and while most of the communication was verbal, caregivers picked up on nonverbal communication, really trying to intimately understand what was being communicated. Caregivers also attributed meaning to these episodes and worked to make sense of these episodes. Overall, these events were largely positive, but there were some negative events. What we do know is that who people were before experiencing dementia, good, bad, and all that showed up during lucid events. Finally, caregivers identified adjustments made to routines or incorporated previous triggers to try to create lucid episodes. Often, mostly, these events were unsuccessful. So here's my contact information. I'm happy to entertain any questions.
Thank you so much, Dr. Cotton. If you have a question, we can take that in the chat Q&A box, or if you want to raise your hand. So go ahead. I have one. Could you tell us a little bit about that interview process with these caregivers that you were quoting there? Were a lot of those quotes from the interviews themselves or from the survey? And what was that process like for you?
Yeah, all of the quotes presented today were from the interviews themselves. I did not conduct any of the interviews. I was provided the data, but I think that from what I heard from the team who collected these stories, and I'm also kind of working on a separate paper regarding ethical and methodological issues interviewing dementia caregivers. What we found is often caregivers were crying as they were telling these stories. People were holding on to these moments for years later. I think that the experience of the caregivers was this interview had cathartic effect for them. I think that our team learned a lot just about the experiences that dementia caregivers are having, and that people were comfortable chatting about these things, whether it pulled on them emotionally or not. I think that folks were excited to share their story, particularly really thinking about helping other caregivers understand the course of dementia. There are certainly, as we sort of kind of take your question and move forward, certainly other things that we have learned and we are thinking about as we go into the R33 and kind of the dissemination phase of the study is like, what's the so what? And so we have tons of thoughts about how we not only interview dementia caregivers, but what do we do with this information? What's the implication for how we train caregivers when these events occur? What types of things may sort of pop up to help caregivers manage these conversations? What type of things might shut down a lucid event? So there are tons of implications for us as we sort of think about dissemination. But yeah, that's the long answer to your question.
Can I ask a question, Hannah? I think so. Hannah, can I ask a question? Oh, I think she's muted.
Sorry, I'm on mute. I'm saying yes, go ahead.
Hi, thanks, Dr. Cotton. That was an awesome talk. I was wondering if you would talk a little bit about the mechanism that's supporting you. I think that might be of great interest to some of our trainees. Would you mind describing that grant mechanism?
Yeah, this was a mechanism that came out of a conversation that was had with NIA folks. There are currently at least 4 active grants that are in the R33 stage at the different sites that have been identified. I do know that when I was at NAC, someone mentioned that there were opportunities for early career investigators to become engaged with the work. And so I actually think that a NAC representative might be better positioned to answer that question, as they've been perhaps keeping in closer contact with the 4 awarded projects. And yes, I would be interested in continuing to conduct these interviews in the future. There is a lot that we have to learn. I think that the foresight, although we have some papers that we're working on together, I think there is an opportunity to pull all of this data together across the site and to really sort of look at this phenomenon in multiple ways. There are tons of opportunities in terms of having conceptual clarity about a lucid event, implications for how we conduct clinical assessments, implications for how we think about personhood, how that shows up in caregiver training and training for persons that reach caregivers. There are all sorts of sort of potential opportunities involved. And so I think that maybe getting in contact with NAC regarding next steps would be helpful. But if anyone's certainly interested in Minnesota, I'm happy to, and Wisconsin, I'm happy to connect you with the folks in the R33 sort of a portion of the study.
Wonderful. Well, we are at time, so thank you so much, Dr. Cotton, and great work. Our next presenter is PhD candidate Meagan Lauber. Meagan is with Boston University and will be presenting work today on graphical model analysis of how global amyloid influences regional tau dynamics in PET imaging.
Does everybody just see loading on the screen?
Yes.
Okay, let me try again.
If not, just make it larger and just go with the other flow.
Yep, we don't mind seeing those. That's
Yes,
fine.
that's
Okay,
fine.
it,
Go
it was
ahead.
working like my try won't. It was literally working like 5 minutes ago. We did a technical test.
We
but,
saw you. You're good. Don't worry.
We can always come back to you too if you want, Meagan, whichever you
Um,
prefer.
Yeah, maybe let me try to figure this out because there's a lot of animations in the slides.
Absolutely. Yeah.
Stop
So
and see.
then let me go to our next presenter.
Surprise, everybody be on your toes.
All right. So our next presenter is Naomi. Naomi, are you ready? Okay, great. So our next presenter is Dr. Naomi Gaggi. Naomi is a postdoctoral fellow at NYU ADRC, and today they'll be presenting on the effects of transcranial photobiomodulation on cerebral hemodynamics and early Alzheimer's disease. Take it away.
Thank you for the introduction and the opportunity to speak. Today I'll be talking about the effects of a neuromodulation technique called transcranial photobiomodulation and how that affects brain activity in early Alzheimer's disease. No disclosures. Alzheimer's disease, or AD, is a disorder of impaired memory and cognition, and there are many brain-related changes seen, such as plaques and tangles, hypoperfusion, aberrant neuronal activity, brain atrophy, and mitochondrial dysfunction. A growing body of literature points to the critical role of impaired bioenergetics in AD involving a dysregulated neuronal metabolism. Impaired cerebral metabolism can even precede the onset of AD. In people with mild cognitive impairment, we can see declines in glucose metabolism and mitochondrial dysfunction. And the mitochondrial cascade hypothesis represents one formal attempt to explain how mitochondria may serve as the primary generator of AD, making mitochondria potential therapeutic targets for AD prevention or early intervention. So there is a non-invasive, well-tolerated, easy to perform, free of significant side effects, and last but not least, cost-effective neuromodulation strategy called transcranial photobiomodulation, which I'll refer to as TPBM. This has the potential to target mitochondria and enhance cerebral metabolism in addition to boosting memory and cognition. TPBM uses red and/or near-infrared light photons to penetrate the cerebral cortex. This method has been done using lasers or LEDs. Once the photons enter through the skull, they are able to pass through the neuronal cell membrane by way of light-sensitive ion channels, depending on the specific wavelength. The photons are then able to enter the mitochondria, which is the main site of physiologic change related to TPBM. More specifically, photons are absorbed within the mitochondria by the last complex of the electron transport chain, cytochrome C oxidase, or CCO. This then results in an increased ATP production, the release of nitric oxide from CCO, and a brief increase in reactive oxygen species. Benefits seen after TPBM include increased cerebral hemodynamics and metabolism with high reproducibility across multiple clinical and healthy populations. And while a growing number of studies are using TPBM in cohorts, including early AD, not many studies have examined the influence of TPBM on regional brain activity and metabolism, which may serve as a modifiable therapeutic target in early AD patients. There is an ongoing collaborative study between New York University, Nathan Klein Institute for Psychiatric Research, and MGH called TRAP-AD, or transcranial photobiomodulation for Alzheimer's disease. And the aim of this study is to determine if the application of TPBM using near-infrared energy can help improve cognition, memory, and cerebral blood flow in people with mild cognitive impairment or early AD. The overview of the study design is that participants come in for screening, clinical, and cognitive assessments, and then they come in for another visit, and this is called the target engagement session. So we have a TPBM device that is MRI compatible, so we put the device on participants' heads, they lay in the scanner, and then we do 3 consecutive scans immediately following a structural T1. So we look at the structure of their brain, then we have a pre-TPBM scan where we look at the bold signal during resting state. After that scan is over, we turn the laser on, so we're simultaneously recording and stimulating the brain. And then we have the post-TPBM scan where we turn the laser off and we're again just looking at resting state. After this target engagement session, participants are randomized into either the TPBM or sham group, and they receive treatment 3 times a week for 8 weeks. And then we look at outcome variables at the end, but this is an ongoing study and we're wrapping up pretty soon. But if you want to learn more about inclusion exclusion or more details about the study, because this is pretty simplified, I put a code in the right corner. But for this talk, I'll just be focusing on the single target engagement session where participants are in the scanner and receive a single dose of TPBM. So we are again simultaneously recording and stimulating the brain, and these are 3 sequential scans from 1 day. The aim of the current analysis is to explore the effects of TPBM on local brain activity in early AD. And how I explored local brain activity is by measuring FALF, or the fractional amplitude of low frequency fluctuations. FALF is a measure of regional voxelized spontaneous fluctuations of the bold signal, which reflects variations in intrinsic brain activity. This is measured between 0.01 and 0.08 hertz of frequency range. FALF values were correlated strongly with regional PET measurements of cerebral glucose metabolism. And because improving cerebral metabolism is an established and well-studied mechanism of TPBM, we wanted to explore if FALF and if this can be engaged using TPBM. At the time of the analysis, we had 19 participants, mean age of 74, and all met Peterson criteria for early AD or mild cognitive impairment. And just revisiting the study design very briefly, we are focusing on the target engagement session. In this analysis, this was 1 day, 1 session, where we have a pre-TPBM, during TPBM, and post-TPBM resting state MRI scan, where we measured bold signal. TPBM was measured bilaterally to standard EEG coordinates, including F3 and F4, which are approximated to be the bilateral dorsolateral prefrontal cortex, FP1 and FP2, which are approximated to be the bilateral frontal pole, and FC and FPZ, which are approximated to be the medial prefrontal cortex. So this is what the MR-compatible TPBM device looks like. The 2 rectangular pieces on the headband, that's where the laser light is entering from, and the device rests flush to the forehead. So this is designed to target the EEG coordinates that I had just mentioned. We created ROIs where the device is projected to irradiate to the cortex using paired t-tests between the pre and during and pre and post conditions. We found that in frontal ROIs illuminated by TPBM, there were significant increases in FALF from pre to during and also from pre to post TPBM. And these regions included F3, F4, FP1, FP2, and FC. We did not see these changes in FPZ. So in each graph on the screen, you'll see that there's 3 time points, the pre, during, and post, and these are the conditions within each ROI. Each line within the graph represents individual changes, and most participants did show a significant increase. However, if you look, there's a gray, orange, and blue line. Those are the same 3 participants within each ROI, and they had a decreased FALF in response to TPBM. So this may indicate some kind of non-response or a different response to TPBM. We did have strong effect sizes for these changes in most regions, including F3, F4, FP1, FP2, and FC. We did not see these changes in FPZ, and this is because we suspect that there is increased noise, signal dropout in this region, and also this may be influenced by the aging brain. Most areas directly irradiated by the laser did show changes in FALF, with Cohen's d ranging from 0.7 to 1.1. So in conclusion, despite a small sample size, effect sizes for the change in baseline FALF in the target regions during and post-TPBM are medium to large. Effect sizes were larger during the TPBM condition versus after. So the takeaway here is that the laser technique is able to engage brain activity in target regions during illumination and somewhat after. We also may have observed an unexpected decrease in FALF in a subset of 3 participants. This difference in response may serve as a predictor of outcome for TPBM. Like I said, we're wrapping up the study, so we hope to look at this soon. But looking at just basic demographics and clinical data, we didn't see anything differing these 3 participants from the rest. Future studies should compare regional changes in FALF to changes in PET metabolism pre and post TPBM. Such work could also inform on the biological interpretation of FALF, which is non-invasive and more accessible. Outcome variables will be correlated with these findings after the study completion. This was just the first stage of analysis. Since I presented my poster, I did a little bit more exploring of this data, and we're nearing the end of the study, so we looked at a little bit more, but there's more to come. So since the mechanism of TPBM is through activating the mitochondrial transport chain, we wanted to see if baseline phosphorus MRS measures can predict the changes in FALF that I had just demonstrated. So before they had come in for the target engagement session, they had an MRS session. And what we found was that FALF from pre to post TPBM with baseline phosphorus MRS measures within the frontal ROIs irradiated by TPBM, including F3, F4, F2, FP2, and FP1 projections onto the cortical surface. So we found that changes in FALF from pre to post TPBM were correlated with phosphocreatine to inorganic phosphate ratio, and changes in FALF from pre to post TPBM were correlated with pH. Areas with decreased oxidative metabolism at baseline indicated by a decreased pH and a low phosphocreatine to inorganic phosphate ratio showed the largest increases in FALF after TPBM. All participants had a decreased pH in those ROIs, so it was less than 7.4, indicating mitochondrial dysfunction. However, those with a pH closer to 7.4 demonstrated a greater change in FALF after TPBM in these frontal regions where neurodegeneration does begin to occur. pH levels will eventually decrease as AD progresses, and they continue to deteriorate. But it is noteworthy to note that the less impaired but still dysfunctional pH did show greater changes to FALF response. So this is just a hint of what may come from the data set. It's a very small sample size, and we will continue to look into these measures and if they can be markers of engagement. And thank you all so much for your time and attention. And thank you to my mentors and PIs of the study, Dana Iosifescu and Ricardo Osorio, the rest of our team, to our funding sources, and NAC and the REC program. Thank you.
Thank you so much. We have time for one question. And if there are no questions, I do see that you said this work has been published and secondary analysis have been conducted. If you have any links to that publication, feel free to drop it in the chat and people can check it out further.
Perfect. Thank you. I'll do that right now.
All All right, great job. It doesn't look like we have any questions coming in at this time, but if any do percolate, feel free to drop them in the Q&A box and Naomi can type the answer to the question there. I think we're going to turn it back to Meagan Lauber. Meagan, how are things going?
Good. Let me try. It should be working. I think it was just some hefty data sheets that were maybe there. Awesome. Okay, great.
Wonderful. Okay, so I will go ahead and reintroduce you. Today we have presenting Meagan Lauber, who is a PhD candidate from Boston University, ADRC, and they're presenting on graphical model analysis of how global amyloid influences regional tau dynamics in PET imaging. Go ahead.
Thank you guys so much for coming to my talk today. So I'm sure you guys are familiar with the 2018 NIA biomarker-based framework of Alzheimer's disease. So this is kind of what I will be focusing on, specifically the interactions between amyloid beta and tau. So there was this really great review paper in 2020 that kind of summarized a lot of literature showing that amyloid beta appears first, but also plays this kind of instigatory and acceleratory role in tau spreading. However, these in vivo dynamics are still not completely well characterized. And then there was also a 2021 paper by Duray et al. That kind of showed in PET imaging that elevated global amyloid was almost like a prerequisite for tau spreading beyond its initial seeding in the medial temporal lobe, and that it kind of played a further acceleratory role in its accumulation throughout the brain. So kind of based on the synergistic relationship between the two pathologies and also just the different patterns of accumulation as seen in this figure, I kind of wanted to examine this relationship a little bit more closely. So specifically, I wanted to examine how regional tau dynamics, defined as how much tau is in an individual brain region, which is measured by something called a standardized uptake value ratio, and how these kind of these dynamics might change at different whole brain global amyloid burdens, which are measured by centiloids, which is a standardized measure of whole brain amyloid pathology. So essentially, to kind of narrow in, the specific aim of my project was to combine machine learning and graph theory to examine how higher global amyloid burden influences the spatial dynamics and efficiency of regional tau deposition. And based on the previous work I read, I expected to observe an increased efficiency and enhanced network organization of tau as amyloid burden increased. So to kind of help you understand like how I structured the analyses, I think it's really important to understand how I selected data and structured my analysis groups. So I had access to data from the ADNI and A4 cohorts. I then selected people that had both a amyloid and a tau PET scan. And then because I was interested in kind of like the dynamics of tau within like already elevated pathological amyloid, I subdivided based on those who met a centiloid cutoff value previously established for amyloid positivity. And then once I had those people who were amyloid positive, I then used a very simple quantile thresholding approach to separate those people into three like quantile, centiloid quantile groups. So I'm going to go a little bit into my model now that I use just to kind of like help clarify and understand, but then I will again like summarize many times, like I'll go back to the science. So I'm sure you guys are all familiar with a covariance matrix. However, the model that I'm using calculates a related thing called the precision matrix, which is essentially just an inversion of a covariance matrix such that a non-zero entry in the precision matrix implies a direct dependency between variables. So it's essentially describing, similar to a partial correlation, how two variables relate when the effects of all other variables are removed or controlled for. So this brings me to probabilistic graphical models, which are a subset of machine learning that kind of learn probabilistic relationships between a set of random variables. And the specific model I use is called a graphical lasso model. And essentially what this model does is it's estimating the sparse precision matrix from a set of data such that any non-zero entry in this resulting precision matrix suggests a direct dependency between two variables. And how it accomplishes this is it uses uses lasso regularization to promote sparsity or enforce sparsity in the precision matrix, such that weak correlations are shrunk to zero. So it's kind of performing this automatic feature selection in that it's applying this lasso penalty so that only when you get the output of the model, only the most significant relationships are remaining. Because the weak ones have been set to zero. So this approach is really handy, especially in biological research, because it reduces the influence of weak associations and noise and removes the effect of like confounding variables in theory. And so kind of like start to finish, the entire analysis framework for my project was first, this is pretty standard for any machine learning or modeling approach, is you want to, if you have a hyperparameter, in my case, I had a hyperparameter for the model of alpha, which is the intensity of the lasso regularization you're applying. So essentially, you plot a range of different hyperparameter values. You plot something called the Bayesian information criteria and find the first minimum. And essentially what this is telling you is for your model and your data, this is the optimal trade-off between model complexity and fit to the data, like goodness of fit. So essentially, once we have the hyperparameter, you can then plug it into the model, and then the model estimates the precision matrix from the set of random variables, which in my case were the regional tau values. Then you interpret it as a graph. And then after you have it in as a graph, you're able to kind of calculate these really cool graph and network science properties and analyze how they change at different amyloid burdens. So again, like to put it all together, I know this is a lot. The whole model schematic from data to output is essentially for each amyloid burden subgroup that I've constructed for each cohort, so 6 total analysis groups. You're essentially taking like the low A4 centilloid group on its own, putting it through this model where the input to the model is each row represents an individual's regional tau PET SUVRs across the whole brain, with each column being a different brain region. And then essentially what you're doing is when you feed it into this model, the graphical lasso model is estimating the partial correlation between every pair of regions across the entire centilloid group. And then when you get this kind of outputted partial correlation matrix or precision matrix, then you derive the partial correlation matrix from that. It's a little technical. Essentially, then the output of that is this matrix with only the most important correlational relationships remaining, because the weak ones have been kind of set to zero and then removed in that way. And so then you can view these as a graph. And so as you can see here, like previously, if you were just to construct a correlational graph, A and C might have been connected, but because that relationship had been set to zero, it does not appear anymore as a link in the graph. So that's how you're kind of doing this like feature selection of only the most salient important relationships. So again, the advantage of this is just the ability to identify direct relationships in regional tau deposition at the population level. And then you can compare this across different global amyloid burdens. So what do I mean by efficiency and organization? So these are some graph theory network science metrics that I thought would be most interesting to kind of interrogate this biological approach with. So essentially, I analyzed shortest path length, which is the efficiency of flow from node to node. Essentially, how many steps does it take to get to here, from here to here? So in a very efficiently organized network, you would have a low shortest path length, meaning you could get from here to here in a relatively few number of steps. Similarly, but kind of on a more like local scale instead of long range connections, the clustering coefficient is this kind of degree of local interconnectedness, such that a high clustering coefficient suggests that the network is highly connected among neighboring node hubs. And then there's the small world coefficient, which is this kind of like holistic overall network efficiency metric, measuring both across long range connections and efficiency across neighboring nodes. And there's been a lot of like graph theory research into like various biological systems, and generally a high small world coefficient kind of indicates this optimal network organization and high efficiency. So here are the results. Something I didn't mention before, but the rationale behind selecting both the ADNI and A4 groups was so that you could kind of have a validation cohort per se, so that you could see if these patterns were replicating and yeah, very exciting to see that they did generally. And I can go back to the slide, but I did prepare like a much more interpretable takeaway from these results. So essentially what we saw was that as at increasing amyloid burdens, the clustering coefficient tended to increase, indicating the tau networks were becoming more locally efficient. The shortest path length decreased, indicating the long range connections in the tau network were becoming more efficient also, and the small world coefficient increased, indicating this kind of enhanced overall efficiency of the tau networks. So what's the clinical relevance or significance of this? Something that I've been really excited about is this kind of prospect of developing combination anti-amyloid, anti-tau therapies, or just tau therapies in general, or even just like trying to figure out what the optimal time is based on someone's biomarker framework to actually intervene with the current existing anti-amyloid drugs. So what I thought was really interesting was we saw this kind of marked increase in tau efficiency when going between the medium and the high amyloid burden groups. And this was, based on thresholds that I determined, so it's not exactly like a hard and fast threshold, but generally like around a centiloid value of 94, which is the bottom end of the high amyloid group, we're kind of seeing this really dramatic increase in tau efficiency. And so my kind of takeaway from that is that any kind of like intervention therapeutic intervention should occur before this increase in tau efficiency happens, because like then you would be able to curb the widespread tau proliferation. And as we know, tau is a lot more correlated with the kind of like functional deficits and memory issues that are characteristic of dementia. So that was kind of something that really was exciting to me. And future directions include kind of like applying this method into different like ATN subtyping to kind of help characterize biomarker profiles. And I just wanted to say thank you to both of my amazing labs, to my graduate program, to the National Science Foundation, GRFP, that supports me, and also to the NIH and BU ADRC for the chance to participate in the REC program. Thank you guys so much. Oh, and also there's a link to the paper in the like the advertisement for this webinar. I can also put it in the chat.
Great. Fantastic work. Do we have any questions that are coming in for Meagan? All right, looks like Meagan put that publication in the chat to me. I will put it out to everybody. Questions for Meagan? So
I have a question, Hannah, if
Yeah,
no one
go ahead.
does. Oh, I keep clicking myself off. There we go. So really great work, Meagan. Can you talk a little bit about how you became part of the REC cohort and how that happened, how you were hanging out doing machine learning, and how is this going to help your career?
So essentially I was lucky enough in that this was the first year they opened up the REC program to PhD students. So it just worked out really well that I was super excited about applying. So my primary mentor, Vijaya Kolachalama, has dual appointments in the BU ADRC within the School of Medicine, but then also the Center for Computing and Data Science. So that's how I'm kind of, in between like the machine learning and neuroscience. But yeah, the REC program was a really like valuable opportunity because it kind of enabled me to strengthen my biological understanding of the disease. I mean, I am a neuroscientist by undergraduate training and by program, but I had not had the time to kind of like hone more of that, like, the biological perspectives and also just like the clinical perspectives. So the REC program was a really good opportunity to be able to get to have like input and talk with a lot more diverse like dementia and Alzheimer's specialists. And it also supported me going to a really cool machine learning workshop where I was able to kind of like learn more about different ways I can analyze the biological data we have.
And win the poster competition. So congratulations. Thank you.
Thank you.
Yeah, really great work. I think we're going to pivot then to our last presenter if there are no more questions. Our next presenter is Dr. Frances Cambronero. Frances comes to us as a postdoctoral fellow at Vanderbilt Exploratory ADRC, and today she'll be presenting on the neighborhood disadvantage is associated with changes in cerebrovascular health among older adults over a 9-year follow-up period. Take it away.
Unmute myself first. So thanks a ton to NAC and the event organizers for inviting me to present my research today. As Hannah mentioned, my name is Frances, and I welcome all questions you might have on this presentation. Feel free to email me afterwards as well if anything comes up. So as a brief introduction, we know that as the burden of Alzheimer's disease and related dementias rapidly increase, there is a dire need to implement holistic prevention approaches, particularly in lieu of a cure. And it's important, increasingly so, that those interventions consider the wider ecosystem of individual, interpersonal, and structural factors that drive worse aging outcomes. Traditional dementia prevention strategies often focus on lifestyle factors that reduce vascular risk factors for ADRD, including health behaviors at the individual level, such as diet, exercise, sleep, smoking, etc. However, newer prevention frameworks have emphasized the role of surrounding environment outside of an individual's direct control in shaping health options and behavior at the outset. These factors include societal level governance and policy, as well as community level environments across housing, economic, social, and service-related systems. Given their upstream nature and the wide-ranging impacts that these environmental factors have, they're considered particularly high-impact modifiable risk factors for Alzheimer's. Ultimately, these environmental factors are thought to contribute both independently and synergistically to the risk of cognitive decline within the broader range of individual biological risk factors, such as genetics, for example. In particular, neighborhood environments reflect a complex interplay between physical, economic, and social environments that can create or limit opportunities for healthy aging. For example, neighborhood geographies can influence access to essential services like quality education and housing, opportunities for physical activity and nutritious foods, and social factors critical for health, such as community support and socialization opportunities. The Area Deprivation Index represents an increasingly popular and novel opportunity to robustly evaluate neighborhood level risk. It's capable of measuring numerous aspects of the built environment and socioeconomics unique to communities, as shown on the right. Overall, this measure combines 17 census-based indicators of housing, income, employment, education, and essential services at the census block group level. So as close to, as granular as we can get to evaluate neighborhood geographies. And importantly, this measure has been originally created by the Singh group and developed by Amy Kine's group at the University of Wisconsin, particularly in the context of Alzheimer's the past few years. But although the ADI has been linked to accelerated cognitive aging, neurodegeneration, and AD core pathologies, its relationship to cerebrovascular health is not well known. This research gap is especially significant as cerebrovascular pathologies like small vessel disease are highly prevalent in aging. And broadly speaking, vascular risk is one of the most modifiable ADRD risk pathways, which the NIH has also identified as a high priority research area. Thus, the pilot data in my research project has investigated the question of whether neighborhood socioeconomic disadvantage, as assessed by the ADI, relates to cerebrovascular damage and dysfunction common to aging. To answer this question, I've used the Vanderbilt Memory and Aging Project, which is the local cohort at the Vanderbilt ADRC, or one of two cohorts. It was launched in 2012. It's a longitudinal study of older adults, and these adults are relatively heart healthy, free of dementia. This cohort study is really designed to look at comprehensive and well-detailed measures of vascular phenotypes and vascular aging, ideally to capture the earliest stage of vascular dysfunction that we know is a very hot topic in Alzheimer's pathophysiology. The current study uses both baseline and longitudinal analyses. The baseline sample is about 326 individuals, and the longitudinal sample is 296. Importantly, like I mentioned, VMAP, the Vanderbilt Memory and Alzheimer's Project, has very comprehensive measures of vascular health, both at the cardiac and the cerebrovascular level. And the reason is because we know, again, that these pathologies are highly comorbid in aging. Specifically, the underlying damage to the vessel wall, as well as the complex cascade of molecular changes that accompany cerebrovascular aging, such as vascular remodeling, blood-brain barrier changes. Broadly speaking, it is relatively difficult to evaluate this construct in humans, but we do have a solid number of measures that have been traditionally used and emerging measures that are increasingly used nowadays. Traditional small vessel disease markers include MRI visible tissue changes of presumed cerebrovascular origin, as well as MRI quantifiable changes in blood flow, hemodynamics, whether that's resting blood flow or reactivity, so more of a functional measure of blood flow regulation. And more novel measures that have been recently optimized and developed have been fluid-based molecular changes reflective of cerebrovascular damage and dysfunction. And specifically in the Vanderbilt Memory and Aging Project, we have a number of 3T MRI-based imaging biomarkers that evaluate cerebrovascular health, and those include white matter hyperintensities as assessed on the flare sequence on MRI, lacunar infarcts, which are reflective of ischemic type damage, microbleeds, which tend to be reflective of hemorrhagic type damage, and that's quantified on susceptibility weighted imaging. And as I mentioned as well, cerebral blood flow using ASL MRI. In addition, we have a number of fluid-based cerebrovascular biomarkers, and these include CSF measures of matrix metalloproteinases, both 9 and 2, which, if you see here on the bottom right, are thought to be reflective of cerebrovascular remodeling at the level of both the capillaries and the penetrating arterioles, as well as CSF PDGFR beta, which is a newer, more novel measure of parasite injury. And for those of you less familiar, parasites are the contractile cells, typically at the level of the capillary vessels in the brain that help regulate blood flow and are also an increasingly studied cell type in the context of Alzheimer's. And finally, we have the ability in combination with our plasma measures to look at the CSF plasma albumin ratio, which is thought to be a proxy measure of blood-brain barrier permeability. And that is reflective of overall endothelial tight junction dysfunction or permeability that occurs at the level of the capillaries. So our analytical plan using all of these very rich and diverse cerebrovascular biomarkers includes both linear regression models with the cross-sectional baseline data that relate ADI to those biomarkers that I mentioned on the previous slide, as well as longitudinal models using linear mixed effects regression models that look at the interaction of baseline ADI and follow-up time on those same cerebrovascular biomarkers of interest, minus a couple that we haven't quantified longitudinally, which I'll mention in a second. For all of these models, the covariates that we used in these included age, sex, race, education, and ethnicity, cognitive status, as well as ApoE4 genetic status. For longitudinal models, we included an additional adjustment for follow-up time. And for CBF models, cerebral blood flow models, we included an adjustment for T1-weighted structural volume. And importantly, across these models, false discovery rate corrections were applied to account for multiple comparisons with the a priori significance threshold of 0.05. And so what we found is for our baseline and longitudinal samples, just to characterize the VMAT cohort a little further, this cohort is as typical of a lot of ADRCs, predominantly white and highly educated. But that being said, the median area deprivation index is 33 with a range of about 1 to 98. And with the area deprivation index, the higher values indicate more disadvantage. And our interquartile range for that metric at baseline is about 17 to 51. So about 50% of our participants lie in that sort of moderate to moderately low disadvantage area, but we do have the full spectrum of values represented. As the title of my talk suggests, the data that we have longitudinally is up to 9 years with a mean follow-up time of 5.1 years. And our project results showed that at baseline, ADI is associated with changes in matrix metalloproteinase 9, which again is a marker of extracellular matrix remodeling, presumably of vascular origin, as well as cerebral blood flow, but did not relate to the tissue-based biomarkers of SVD, small vessel disease, that are more traditionally used. Longitudinally, the MMP9 associations persisted, but instead of cerebral blood flow, we saw associations with our parasite marker, the CSF PDGFR beta marker. We did see somewhat not below significant threshold, but modest associations with the Q-albumin ratio, which again is a marker of blood-brain barrier permeability. But again, this did not meet our significance threshold with the current sample size that we have. So to add a little bit of detail about the directionality of these results, at baseline, what we saw was higher ADI, meaning more disadvantage at the neighborhood level, related to lower MMP9, as well as higher cerebral blood flow on the right. Longitudinally, we saw that higher ADI, more disadvantage, related to increases in MMP9, as well as declines in PDGFR beta. And so in summary, to interpret those results a little further, longitudinal results seem to suggest that neighborhood disadvantage may promote cerebrovascular remodeling, specifically via the MMP9 pathway. And we know that MMP9 associations are also reflective in the literature of demyelination and neurodegeneration processes. So the degree to which this is specific to the cerebrovasculature remains to be investigated. It is commonly characterized in blood-brain barrier dysfunction studies, though. Our PDGFR beta results, which again are reflective of parasite injury, were in unexpected directions. And there is some mixed literature on how those PDGFR beta increases might relate to either later stages of neurodegeneration when parasite integrity is close to depleted, or medication effects that might be protective and may serve as confounds, which we haven't looked at. Cross-sectional results, like I mentioned, suggested that cerebral hemodynamics, cerebral blood flow, may be implicated at stages prior to enrollment. So for example, midlife, or potentially those results could arise from confounding effects. So those pose future directions as well. Overall, findings support the hypotheses that adverse environmental conditions may contribute to cerebrovascular changes among older adults. And the data overall provides more evidence to the growing body of literature that suggests that community development initiatives, some of them are illustrated on the right here, are integral to health. So instead of thinking them as sort of separate constructs, it's great that recent literature has started to integrate these more and thinking more holistically about how not just social determinants of health, but broader environmental influences across different industries or segments of society contribute to worse aging or better aging if we're talking about protective factors. And overall, we're excited with these pilot data to further incorporate a second wave of recruitment that we have recently finalized. So the power will be greatly increased in those follow-up analyses that I'm currently pursuing. Future studies should investigate temporal effects, meaning at different stages of the life course, or at least in older aging, how these associations might vary, as well as mechanisms through which ADI may actually impact vascular health. Again, I mentioned at the very beginning that ADI is an upstream factor that is highly related to health behaviors, lifestyle factors, and obviously it's a composite measure. So we have some ability to look at how the individual measures might be driving these associations. And broadly speaking, we know that ADI is intertwined with broader ranges of sociodemographic factors. So looking at interactions with these factors and how these associations might vary depending on different identities will be of utmost importance as we continue to diversify our cohort as well. So I'd like to thank our team at the Vanderbilt Memory and Alzheimer Center, as well as my mentor, Dr. Angela Jefferson. And also a big thanks to my training funding sources over the years listed on the top right, as well as our center funding sources. And I'll take any questions that you all might have at this time. Thank you.
Wonderful work. Thank you. It does look like we have one question in the chat. They say, great talk. I might miss it, but were the CSF results controlled for individual medical conditions such as presence of diabetes or hypertension?
Yeah, that's a great question. So these analyses, both the baseline and longitudinal ones, adjusted for the Framingham stroke risk profile, which is a composite of vascular risk, which includes, like you mentioned, diabetes, hypertension, as well as I think about seven other measures of cardiovascular risk. And that's an important factor to control for sure. So thank you for bringing that up.
This was awesome. Thanks, Hannah. Thanks, everybody. Any last words,
just
Hannah?
Ask one final question. Francis, would you mind telling us about your experience as a rec poster session presenter and maybe some of the connections that came out of that?
Yeah, I talked to so many people, and I think about this project is currently being written up into a manuscript, which I'm hoping to submit in the next couple months. I'd say about maybe 25%, if not more, of the revisions that I made were based off of that poster feedback session. So I received a ton of really useful comments on additional considerations, limitations, and it really helped me not just understand how to communicate my science across disciplines, depending on people's backgrounds, but important considerations that I wasn't even at the time considering. So it's really been a pleasure to participate in that program, and I appreciate all the support that NAC has offered over the past year and a half.
That's great. Well, good luck with that submission. I think we are going to call it smashing success on this first Rec Rising Star Awardee Research Showcase. So thank you all presenters and Rec Steering Committee members for helping organize this event. We will be posting this recording on our YouTube channel, so feel free to share this research with others who might not have been here today. And then we're really excited to have another Rec poster session opportunity at the upcoming spring meeting in May, and that will be in San Francisco. It'll be an in-person only poster session, but we'll be accepting nominations for that shortly. So if you are a Rec scholar who's interested in presenting your work, make sure you talk to your Rec leader about your interest in potentially presenting there, or vice versa. If you are a Rec leader, make sure to be talking to your scholars and see where some of that interest may lie, and we'll be collecting nominations shortly. Any other final notes from our Rec Steering Committee?
I think it was, I agree, Hannah, smashing success, and we look forward to seeing hopefully many of you at the spring meeting at your poster session. Good luck.